Hi, everyone. Last lecture, we focused on the plasma membrane as a boundary. It separates the inside of the cell from the outside world and helps maintain the conditions required for life. But that creates an important problem. Cells are not closed systems. They constantly need to take in nutrients, water, and other molecules from their environment. They also need to remove waste and maintain proper internal concentrations of ions and chemicals. So the membrane has to do something that seems almost contradictory. It must create a barrier while still allowing controlled exchange with the environment. And that is where membrane transport becomes critical. In this lecture, we'll explore how substances move across membranes, why some molecules cross easily while others cannot, and how cells actively regulate transport to maintain homeostasis. By the end of this module, you should be able to differentiate between ways that substances move across the plasma membrane. One of the most important ideas in membrane transport is diffusion. At the molecular level, molecules are constantly moving. They collide with each other, bounce around randomly, and spread through available space. Because of this random motion, molecules naturally tend to spread out over time. If one region has a high concentration of molecules and another region has a lower concentration, there will be a net movement from the area of high concentration towards the area of low concentration. This movement does not happen because molecules are trying to go somewhere. It happens simply because random motion makes spreading out more likely than staying clustered together. As molecules spread, they create what we call concentration gradients, differences in concentration across space. Diffusion is extremely important in biology because cells constantly rely on concentration gradients to move substances into and out of the cell. One way substances move across membrane is through simple diffusion. Simple diffusion is a passive process, meaning the cell does not need to spend energy to make it happen. Instead, molecules move down their concentration gradient from areas of higher concentration to areas of lower concentration. Some molecules can pass directly through the phospholipid bilayer because they are small and nonpolar. Nonpolar molecules interact more easily with the hydrophobic interior of the membrane, allowing them to diffuse across relatively easily. Two important examples are oxygen and carbon dioxide. Cells need oxygen for cellular respiration, and they produce carbon dioxide as a waste product. Because both molecules are small and nonpolar, they can often move directly through the membrane by simple diffusion without needing transport proteins or ATP. Osmosis is a special type of diffusion that involves water. In osmosis, water moves across a selectively permeable membrane from areas with lower solute concentration toward areas with higher solute concentration. Another way to think about this is that water tends to move towards the side with more dissolved substances. This movement is extremely important because water balance strongly affects cell structure and function. If too much water enters a cell, the cell may swell and potentially burst. If too much water leaves the cell may shrink and lose normal function. Because of this, cells must carefully regulate water balance to maintain homeostasis. Different organisms have evolved different strategies to deal with osmotic stress, especially organisms that live in environments with very different salt or water concentrations. Not all molecules can cross the membrane through simple diffusion. Many substances are too large, too polar, or too highly charged to pass through the hydrophobic interior of the phospholipid bilayer on their own. In these cases, cells use facilitated diffusion. Facilitated diffusion is still a passive transport process, which means substances move down their concentration gradient from high concentration to low concentration. However, facilitated diffusion requires membrane transport proteins. These proteins create pathways that allow specific molecules or ions to cross the membrane more easily. Importantly, the cell does not need to use ATP or cellular energy for facilitated diffusion because the molecules are still moving down their natural concentration gradient. Facilitated diffusion is especially important for transporting larger molecules and charged substances that cannot move directly through the membrane by simple diffusion. In active transport, this is a a completely different process that requires energy to move substances against their concentration gradient. These transport proteins are going to allow nutrients to accumulate on one side of, or usually on the internal side of the plasma membrane. And to do this, they must use ATP to move those large molecules across the membrane against their concentration gradient. This is different from facilitated diffusion. Membrane transport is not just a small cellular process, it is essential for survival. Cells must constantly take up nutrients from their environment to build biomass, grow, and carry out metabolism. At the same time, they must remove waste products that could become harmful if they accumulate inside the cell. Transport also helps cells regulate salt and water balance. If cells cannot maintain proper ion concentrations, normal cellular function quickly breaks down. Membrane transport is also involved in cellular communication because many signaling molecules and ions must move across membranes for cells to respond to their environment. And in many microbes, membranes play direct roles in energy generation. Important energy producing processes occur across membranes using concentration gradients and transport proteins. So the plasma membrane is not simply a boundary, it is an active interface between the cell and the environment. Now that we understand how substances move across membranes, we can zoom back out and look at another major feature of cells, their shape. In the next lecture, we'll explore cell morphology and arrangement and how differences in cell shape influence microbial function and survival. With that, thank you so much for attending this lecture, and I'll see you at the next one.